everyone. My name is Gongjiang from China. I have to thank the organizer very much to give me this opportunity to present our recent work on the hidden human proteome, the protein-coding, non-coding RNAs. As far as we all know, approximately 2% of the human genome are annotated as protein-coding genes. So there is a question, are all the rest 98% of the non-coding part of the genome are really non-coding? Or, in other words, are there any proteins coded by the non-coding genes? So here we have to define the proteins means that the polypeptides larger than 50 amino acids. We first found that approximately 1,300 non-coding RNAs are being translated in human lung cells. And in that study, we found this, and this triggers our interest on searching for the protein-coding link RNAs. And we searched in the literature. We found some examples sporadically coming from year to year. For example, this ESRG is still annotated as non-coding RNA in the NCBI database. You see this NR starting accession number means that it is a non-coding RNA. However, in 2012, a study, it's here. In 2012, a study showed that the non-coding RNA ESRG was found to encode HESRG protein, and they have very solid evidence for that. And it is even a biomarker for intracranial germinoma and embryonal carcinoma. So, the protein-coding non-coding RNA are really there, and there can be a lot of them. So we performed RNA-Seq or translatom sequencing to see how many link RNAs can be translated in various cell lines. As you see here, we tested 9 cell lines and found 1,000 to 3,000 translating link RNAs in each cell line. Altogether, we found more than 5,000 link RNAs can produce proteins that are larger than 50 amino acids. And we found that there is almost no chromosome specificity. This indicates that this phenomenon is universal across the human genome. Well, next we performed the ribosome profiling to see that we see the moving ribosomes along the predicted ORF. So we are further sure that there are proteins can be produced, not just the ribosome stuck in there on the mRNA. And analyzing these 9 cell lines, we found out that the expression pattern of the new proteins showed clear cell or tissue specificity, shows that the new proteins are not randomly translated. So all of these things indicate that there is really a large hidden human proteome that we have never been discovered. So we have several mission objectives. The first one, we have to find the new proteins, not just in the translation level, but in the protein level, in genome-wide scale. First, we see that the predicted new proteins are generally very short, much shorter than the PD-1 proteins. So this is a big challenge to the mass spec detection. Therefore, we decided to use mass spec in 2 batches. One is a normal whole cell lysate protein mass spec, and another one is that we enrich first the low molecular weight proteins and then put these proteins into mass spec. Of course, we have to follow the criteria, at least 9 amino acid peptide lengths and 1% FDR protein at protein level. So very briefly, we detected in together more than 300 new proteins. So in the total protein mass spec, we take so many, more than 200, and in the low molecular weight fraction, we detected 92 more. And you see that the intersection is very small, showing that the enrichment in low molecular weight protein, this strategy really works. That gives us much higher efficiency in detection. And we see how many unique peptides are there for each new protein. There are 47 supported by at least 2 unique peptides. 24 are supported by 1 unique peptide plus 2 supportive peptides. 42 are supported by 1 unique peptide plus 1 supportive peptide, and the rest, 195, are supported by only 1 unique peptide. But this is quite understandable because in general, the new proteins are pretty short. So it's very hard to have multiple unique peptides that are detected in mass spec. Secondly, we checked manually all these spectra, mass spectra, and most of them are pretty nice. This is just one example. You see continuous Y ion identifications for this peptide. And we validate these new proteins using MRM, of course not all of them, but many of them. This is an example of MRM validation. We also use raised antibody for these new proteins. As you can see, here are some examples that we validate these mass spec detected new proteins using antibody in human cell lines, and these are human tissues. Also, there is an in vitro translation products as positive control, and these two are the whole proteins from 2 different bacteria, so as negative controls. So we are sure that our identifications are true. Also, there are a lot of new proteins that are undetected by the mass spec. For these, we randomly chose several of them and raised antibody. As you can see, we can validate all of these by Western blot. So there are some limitations for the current mass spec instruments. So if in the future, if the instrument develops, we may detect more new proteins in mass spec. Okay, so the next mission objective is, are these new proteins mistranslated or functional? Well, to answer this question, actually, it is not very easy. So we, in the beginning, we proposed a translation ratio, or TR. This is calculated by the RNC mRNA level and divided by the mRNA level for each gene. In the previous study, we have shown that this TR value reflects the translation initiation efficiency, and higher TR means more active function in the cell. So we investigated on the TR values, TR distributions for the new protein against the PE1 proteins. As you can see, in all these cell lines, the distribution, in most cell lines, the distribution of the new protein has, in general, higher TR values than the PE1 proteins, which means that the new proteins are in general more efficiently translated than PE1 proteins. So if this is true, we have we must have some other evidence for that. And we checked the RNA folding energy near the ATG, or the starting site, translation starting site of the new proteins and the P1 proteins. As you can see, the new proteins have less stable mRNA secondary structure than the P1 proteins. So this is an evidence to explain that these new proteins are easier to be translated. Since people have never realized the existence of such new proteins, of course, no one knows what does it do, what the function of these. But we can predict the localization of the new proteins using computational methods. So in the computational methods, we see that the new proteins are enriched in membrane, nucleus, and mitochondria. In general, they have clear localization. So we fused many of the new proteins with EGFP and confirmed their localization exactly as the prediction told us. Here are some examples, and here are more examples. So, the new proteins should be functional because they are stable in full-length form, they have higher translation efficiency, and they have clear localization. So let's see an example whether it's really functional or not. Here is one example, UBP1-SD6. It localizes in nucleus, so we proved this, and further proved by Western blot. And the knockout of this UBP1-SD6 makes the cell line proliferate lower and have decreased wound healing ability. So we have actually ongoing projects for 5 to 6 new proteins, and they are all functional. So we can have a general conclusion that the new proteins are functional. The third mission objective is why the new proteins were not considered before. First excuse is that these new proteins are lower in abundance in transcription level, translation level, and in the protein level. So, of course, it's harder to detect. Second, it has a higher PI value, so it is more alkalic. It is also more difficult to be detected in the mass spec. Third excuse, that the new proteins are generally shorter, so they have less trypsin digestion sites as here compared to the P1 proteins, so that it has much less opportunity to be detected. Computationally, we see that new proteins are mostly single exon transcripts. As for the P1 proteins, more than half of them are multi-exon transcripts, so it is pretty large deviation. So the algorithms will mistreat them. And we found that the new proteins are not conserved during the evolution. You see that the new proteins, we blasted these new proteins for all organisms along the evolution. And you see that most of them emerges just from the primates, and they have no analogs in previous organisms. But for the P1 proteins, we see that at least you can find some homologs in previous or lower organisms. So this is also a big deviation. Also, we try to find the origin of these new proteins. We see that there are some fraction of them are orthologs of human PE1 proteins. 89 of them are new splice variants of human PE1 proteins. Only a small fraction of them are orthologs of bacterial or viral proteins. And almost half of them, we have no idea where it comes from. So this is also a very interesting topic that should be further investigated. So as summary, there are thousands of link RNAs are being translated into new proteins. We found protein evidence for considerable fraction of them in human cell lines and tissues. These new proteins are stable and functional. These new proteins deviate from canonical proteins in various features. So in sum, human genome should be systematically re-annotated. So if you are still struggling with coding potential of linker RNAs, no more guessing. From now on, you can do the translatome sequencing and immediately you'll find whether these link RNAs are translatable or not. So the RNC mRNA analysis was proposed as the fourth resource pillar in C-HPP. So thank you for your attention. I have to thank our link RNA hackers and co-supervisors, Professor Tong Wang and Professor Qingyu He. Yeah, thank you for your attention.